Hi everyone, this is Mrs. Mancuso, and this is an AP Biology video on inheritance. So we're going to be talking all about passing on your traits. So first of all, I want you to think of this piece of DNA right here. This is one chromosome. And within a chromosome, you could have hundreds to thousands of genes. So gene is just one segment of DNA that codes for one specific protein. So segment of DNA with a specific sequence that codes for a specific protein. And as we go through all of this lesson today, it might be like, oh, it's coding for the length of your finger or the color of your fur or whatever trait that we are studying. That all goes back, the protein production always goes back to the genes. All right, so example. You've got hemoglobin gene coding to build the hemoglobin protein for your red blood cells. One set of chromosomes, each with hundreds to thousands of genes. So again, some of this I'm saying, I'm going to have it all written here, so you can always pause and make sure that you get all of these things written down into your notes. Each gamete, so a sperm and an egg, is each going to have one set. It's haploid, and it's going to have one allele for every different trait for that species. You put them together, and that's going to be your genotype. So I have a lot of vocab in here. All right. Fertilization, so the fusing of the sperm and the egg, is going to give your zygote, your new diploid cell, its genotype. So it will have one of each allele. One came originally in the sperm, one came originally in the egg. All right, here is just a blast of vocab for you. I'm sure that most of it is a refresher, but make sure you really understand each of these. So your genotype is your allelic pair. So right away, I want you to just think about, these are the letters. So if your genotype is big A, then you are homozygous dominant. It means you have both forms of the dominant gene. All right, homozygous, same form. Dominant allele, that's the capital letters. Those are the ones that are most likely to be expressed. The recessive only get expressed if you are homozygous recessive. So big A, you'd be homozygous dominant. Little a, you'd be homozygous recessive. And if you are heterozygous, you have both forms of the allele. All right, on to the phenotype. So that was our genotype. That's what our actual alleles are. Phenotype is how you express those alleles. So if you are big A, will you have brown fur? Or if you are, I guess if we're talking about us, brown hair. Or if you are little a, will you have blonde hair? So it's what the allelic pair codes for. That's your phenotype. How is it expressed? All right. Now I'm going to go through, and I'm going to do a lot of crosses as I go, so I'm sure you remember what a Punnett square is. I'm going to be doing a lot of really quick Punnett squares just to show you each of the different types of inheritance. So this first one, dominance. A dominant trait is exhibited. So if I cross, let's say I'm crossing this and this. So I'm crossing, this is called a monohybrid cross. So I'm crossing two hybrids. Hybrid is another word for heterozygous. When you do this, the first thing is we have law of segregation of alleles. That means we have to separate the alleles. So I'm going to put this parent here and I separate the alleles because that's truly what happens in meiosis. Only some of the sperm are going to get this big A and only some of the sperm are going to get this little a, half and half. Now I take this other parent and I separate that parent. So half of the eggs are going to get the big A, half of the eggs are going to get the little a. And now you have all your possibilities. So 25% would be homozygous dominant. And over here, so I always carry down and across, down and across. And you always put the capital letter first if there is one. So 50% are going to be heterozygous or hybrid. And then 25% are going to be homozygous recessive. So this cross actually showed us several things. It showed us that you can have two parents with dominant traits, so the phenotype is dominant, but you could have children that express a recessive trait, and that's because both parents here are carriers. All right, if I was looking at phenotype, dominant is expressed, so 75 would come out brown, and only 25% would come out white. So if this was something for like fur color, let's say I cross two brown mice. I get the two browns, and from their offspring, so when I look at all of their baby mice, 25% of their baby mice were white, showing that they were both heterozygous. But the dominant trait comes through, which means that if you have heterozygous, it is expressed as a dominant. Okay, moving on to the next one. Incomplete dominance. All right, so this is when you get a third phenotype expressed when you have a heterozygous condition. So let's say I am crossing, sorry, undo. Okay, so now I'm going to say that this is red and this is white. So those are my two alleles. So if I cross a red and a white, I would expect to get 100% red. And if it's incomplete dominance, I do not get 100% red. I get 100% pink. So it is not a combination of red and white. It is a new color. They are not red and white flowers. They are pink flowers. So if you have a certain species that has incomplete dominance, you get a third phenotype. So a phenotype was red, a phenotype was white, and my third phenotype is pink. And I could keep this going, and I could cross a pink and a pink, and I would get 25% red. These two would be big R, little r, so I would get 50% pink, and I would get 25% white. All right, so that's an example of incomplete dominance. All right, next one here, codominance. So there are other flowers. Flowers are a great example of this because they're very easy to follow. They're inexpensive. You can think back, it's what Mendel studied first. And it's very clear to see from their offspring what got passed on. So with codominance, let's say R will give me red flowers and W will give me white flowers. And if I have an RW offspring, they are both red and white flowers. So I'm sure you've seen some kind of flowers that have petals that are both red and white together. Not pink, they're red spots and white spots, both in one. Another great example is blood type. If you have AB blood, you actually have, they use the capital I. It stands for immunoglobulin. It is your blood cells. Well, it's like the blood proteins. But you can have an A and a B, which would make you have AB blood. So that's another cool example of codominance. You could have two dominants for one trait. All right, next one, polygenic. So that would mean that when you do your cross, you're not just looking at one gene or one pair, you're looking at multiples. So like if I have A, B, B, cross with A, B, B. So a great example of this is hair color. And you know it because there's so many different possible phenotypes. It's not even just due to two, it's due to multiple allelic pairs. So if you had to do this cross, you have to do a Punnett square with way more boxes. So let me just show you something real quick. If I was going to do this cross, the rule is you always have to separate the alleles. So if I take this parent right here, I come up with all the combinations of their gametes. So the gamete could have a big A and a big B. I can't give it two A's or I can't give it two B's because the alleles separate and I have to have haploid gametes. All right, so that's a possibility. This is a possibility. This is a possibility. And this is a possibility. 4 possibilities for this parent. All right, so when I do my Punnett square, I am going to have those. Now I go to my other parent. There's only one possibility for this parent, a big A with a big B. So I actually only need to make my Punnett square have 4 boxes. That's all I need. There's no need to make 16 boxes if you're going to repeat the same thing over again. All right, and so then I would go through and I would get 4 different possible genotypes. I would get this, I would get this, I would get this, and I would get this. Okay, and so those are my possibilities, and based on what the A's and B's stand for, I could figure out the phenotypes. All right, next one, linked genes. So when you are looking at one chromosome, so if this is my one chromosome, linked genes means that you have 2 genes on the same chromosome. So a great example is freckles and red hair. So it's really common to see people with red hair have freckles because they're actually inherited on the same chromosome. And when we study fruit flies, we'll look at some other ones, some other traits that are inherited on the same chromosome. All right, if something is sex-linked, that means that the gene that you're studying is on the X or on the Y chromosome. So some great examples of that, hemophilia, oops, I spelled that wrong, philia, and colorblindness. They are both examples of a trait that is sex-linked. So if I wanted to draw that, if I'm a male, I'm XY, so I only have one X. If I have a little H on there, I have hemophilia. If I'm a female, I have 2 X's. So in order for me to have hemophilia, I would have had to have inherited both little H's. That means I had to have a dad with hemophilia. You cannot be a daughter with hemophilia if your dad does not have it. All right, because you have to get that little H from your dad. You didn't get a Y from your dad, so your dad would have to have that trait. Or you could do the same thing for color blindness. I don't like to use C's because I like to use letters where you can obviously tell an uppercase versus a lowercase. But we're going to be getting into studying pedigrees and looking at family trees. Actually, this is a perfect time to show one. So let me just add a little page here. Okay, a circle is a female, a line is a marriage, a male is a square. So there's a husband and wife, and here are their kids. So I'm going to show it like this, and I'm going to show that this daughter has this trait, and this son, whoops, sorry, this son does not, and this daughter does not. So my first question would be, could this be sex-linked? And we just talked about this. If this was sex-linked, she would had to have gotten both forms of those genes from each one of her parents. This, I don't know why it keeps doing that. This male, this is a male, so he's an XY. He would have to have a little H. If he had a little H, he'd have to be filled in. So that right away tells me no, 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 no. Can't be sex-linked. So instead, I say, you know what? This girl simply has an autosomal recessive trait. So autosomal means carried on a chromosome that's not a sex chromosome, and it means that both of these parents must be carriers. They have to have a big A because they're not filled in, so they're not expressing the trait, but they gave her these little A's, so I know that. Now, these 2 kids, they could be big A, or they could be little a, big A, little a. And I do not know that unless they are crossed with a, okay, so I'm going to make this daughter married to a man that is little a, little a. Okay, and now I'm actually going to consider them mice, not people, because this just wouldn't be ethical. So I'm going to say that these are mice, and that this male mouse has this trait and the female doesn't. And so I want to look, this is called a test cross. I want to look, and I'm going to actually look at many, many offspring. So that's why I'm choosing them to be mice, so that I can have many offspring. And so this would keep going and going and going and going, and so many offspring. Now, if any of these offspring came out like this, I have my answer. I know this parent here had to be big A, little a, because the only way you could have a little a baby is if you are getting one little a from each parent. If instead I had 100 offspring and none ever came out with the trait, I would be pretty confident then that my female here would be homozygous dominant. Okay, so again, that is called a test cross. When you test and cross it with a homozygous recessive to find out what your allelic pair is, what your genotype is. Okay, next type of trait, epistasis. So epistasis is when you have one gene that can actually affect the expression of another gene. A great example is pigments. So if you do not have a pigment gene, then you will end up an albino. And it does not matter if you have the gene for black hair, red hair, brown hair. You can't express the gene because you can't make the pigments. So the pigment gene is affecting the expression of other genes, the expression of pigments in your skin, pigments in your hair. And so turning other genes basically off because they can't be expressed because they don't have the pigments. All right, and then the last inheritance type I want to talk about is pleiotropy. Pleiotropy is when one gene could actually end up having multiple effects on the body. So a great example of this is sickle cell anemia. So sickle cell anemia leads you to have blood cells that instead of being red blood cells that are nice and puffy, they have this sickle shape. Well, that is going to lead to lower oxygen levels, and that is going to lead to not being able to like catch your breath when you're exercising, or fainting, or so many different side effects. Or another great example is diabetes. When people have diabetes, they're unable to process sugar properly. And because of their inability to process sugar, it actually affects many organs. It can affect your liver. A lot of people, it affects their legs. Some people actually even have to have their legs amputated because they've had so many circulation issues. It can affect your kidneys. There are many different organs that are affected by diabetes. So that's a great example of pleiotropy. So this prefix, pleiotropy, that prefix, PLEI, actually means plural. So multiple effects caused by a single gene. All right, next I just have this little slide of Mendel, because Mendel was really the forerunner in the study of inheritance of traits. So he was a monk, and he really studied pea plants more than anything else. So here's some things that he looked at. He was looking at seed shape, seed color. We probably use this more than any other when we're doing crosses, or the purple and white flowers, pods, their color, their shape, the flower position, if it's on the end or if they're going out to the sides. These were all traits that he looked at and compared over generations. We now would do a chi-square of that analysis as well. We'll be doing plenty of those. All right, some words that are used when you're doing crosses. Your P is your parents. Your F1 is the first generation. The F really stands for filial, and that is your offspring. And then F, so you can think of them like your children. And then F2, you're looking at your second filial. So if I do a monohybrid cross, first thing, all right, let's say I have, let's do the green and the yellow. So he used P's a lot, and as long as you can make your uppercase and your lowercase look different. So actually, no, I'm not going to use P's because I know mine don't look very different. So, all right, I'm going to say big G is green, and this is the seed color, and little y is yellow. Oh, I'm sorry, little g is yellow. Sorry, you got to have the same letter. All right, so if I do a monohybrid cross, that means these are my two parents. Okay, so I'm going to put a P right here to show that is my parent. All right, I do the cross. So I put this, I separate my alleles, I separate my alleles. Okay, my results are 25% big G, and 50% 25% big G, little g, and 25% little g, little g. Those are my genotype frequencies for my offspring. I also could look at phenotypes, and my phenotypes would be 75% green, because both of these are going to come out green, and only 25% yellow. All right. Usually when we're doing the P and the F1 and the F2, we have to cross two homozygotes. So I'm crossing a green and a yellow. That's my parents. My F1 will be all big G, little g. And so in order to get my F2, I have to cross two of these. So big G, big G. I'm sorry, big G, little g, big G, little g. Okay, now I have my F2. And my F2 is going to be those that I just wrote out. I'm going to have 25% big G, big G. I'm going to have 50% big G, little g. And I'm going to have 25% homozygous recessive. Okay? So there we go with an example of the P, the F1, and the F2. All right. A dihybrid cross. Okay, you're going to need some room for this one, so make sure you give yourself some space. All right, so a dihybrid means I have to have two traits, and I have to be a hybrid for each. So here is my dihybrid cross. All right, so now I have to write out all the possibilities of the gametes. So I could have this, I could have this, I could have this, and I could have this. All right, oops. And now this way. There. Sorry, this is a little big. Okay. Woo! All right. Now I got to write down what these things equal. So I'm going to say big A is green and little a is yellow, and this is for seeds. And then I'm going to say the flower color. I'm going to say this is going to be purple flowers, and this is going to be white flowers. Okay, all right, so there's my possibilities. I'm looking at two different traits. That's why it's di. And it's hybrid because I had each parent be a carrier. So instead of filling in all of those boxes, what I do is I make up a key. And I come up with all my possibilities. So my first one, I'm just going to make a filled in circle. And that is going to be for all of the ones that have green seeds and purple, P-U-R-P-L-E. Okay, purple flowers. Green seeds and purple flowers. So in order to have green seeds and purple flowers, all I need is a big A and a big B. So I'm going to go up to my cross. And again, I'm not filling in all those letters. There's no need to. All of these down are going to have a big A and a big B. All of these across are going to have a big A and a big B. All right, does that make sense? Everyone across here is going to get a big A and a big B. Everyone down here is going to get a big A and a big B. Now this one, nope. Here we go. This one also is getting a big A and a big B. This one's going to have a big A and a big B. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 ended up green seeds and purple flowers. All right, now I make another shape. So I'm just going to make an open circle. And that's going to mean that I have a green seed, but a white flower. So again, I just make up symbols for each of my possibilities. And now, oh, what do I need for a green seed and a white flower? I have to have a big A. It doesn't matter what the second letter is, but I have to have a big A to be green. And then to be a white flower, I have to have 2 little B's. So I go up here and I check it out. All right, there is green with a white flower. There is green with a white flower. Let me just see where else. Sorry. Okay, this one right here. This is going to be green with a white flower. So I have 3. 3 out of 16 are green with white flowers. All right, now I'm going to do a square. I'll do a filled in square. That's going to be a yellow seed and a purple flower. So what do I need to be a yellow seed? I have to be little a, little a. And to have a purple flower, I have to have at least one big B. So I'm going to go up and I'm going to find those in my diagram, in my Punnett square. All right, this one, and this one. So I have another 3 out of 16. All right, and then finally, there's only one box left. There is only 1 out of 16 that is yellow seeds, white flowers. Okay, the only way of getting that is to be all homozygous recessive, 1 out of 16. There it is. So I know you probably haven't done it like this before, where you maybe have so many boxes or that you use little symbols instead, but it'll save you quite a bit of time. What I want you to notice is this ratio. This ratio of 9 to 3 to 3 to 1 is what you get if you have a dihybrid cross with both complete dominance. So both of them showed complete dominance. You were either green or yellow. There was no in between, okay? If instead we had something with incomplete dominance, you'd have to add, there would be 2 more numbers in there of one of the traits, because you'd have to account for like pink or whatever other color there was. Okay, we already talked about test cross. All right, I want to do a quick little talk about fruit flies. This will be the end of my video. Sorry, I know this one is a long one. But I'm getting in fruit flies so that you guys actually get to have this experiment. Fruit flies are just really great to work with. It might seem like, oh my gosh, I'm going to work with these little bugs, but that's actually awesome because you keep them in a little tube. It almost looks like a test tube. They're inexpensive. They reproduce quickly. You can follow the traits generation after generation. Easy to take care of. You're going to learn how to feed them, knock them out, all that sort of stuff. They have very easily noticeable traits. So I put this little picture down here to show like, oh, this is wild type, like that's normal. But instead they could have all these different eye colors, white eyes. Their wings could be vestigial, which means they don't grow in right, or their wings could be curly, or they could have different eye types. There's a lot of different traits to look at, and you can tell right away if they have them or not. They actually only have 4 chromosomes, so you can study linked genes quite easily. This next slide, I just have one more diagram to show. So like having a yellow body and white eyes are on the same chromosome. So you can actually study linked genes, and you can study crossing over frequencies of linked genes. You can also extract from their mouth. In the larval stage, you can extract their chromosomes and do a spread and actually look at it under the microscope. They have very large salival chromosomes. It's really wild. So we're going to be doing all of that. So again, I apologize, such a long video, but I really just wanted to expose you to a lot of different things about inheritance.